Hello and welcome to screencast part 1 for the nervous system. I'm going to try and keep these a little shorter so they're more bite-sized nuggets. Hopefully you've downloaded the latest version of the PowerPoints from OneNote. I've made just a couple of changes. First up is a little bit of historical context just about neurosurgery. The earliest evidence of any sort of neurosurgery goes back to 3000 BC, where there is some evidence of trepanation in China. This is a skull that actually comes from Peru, so that would have been the Incan Empire, as I understand it. And so trepanation was just drilling little holes in the skull to release pressure if people had migraines. I'm not sure how effective it was, but it was medicine, and as you can see here, it goes way back to prehistoric times. So there's also documentation of neurosurgery of different types in Egypt, again in that papyrus that also mentioned the I think it was breast tumors in Egypt as well. This map here comes from this article. This is a link to it in SharePoint. So you have to be logged into the RVC system for these two links to work. But it does show all the other places where there was neurosurgery in addition to just Europe. This would have been the Incans. In Peru would be down here. I forget what's there modern day. Maybe Bolivia. My geography's not very good. And then China's here, Sinai Peninsula, Egypt. And I added a little purple right there for India because there was early neurosurgery in India as well. I forget which of the articles that was in. Moving right along then, onto the nervous system itself. We're not going to talk about all the different things that the nervous system does to participate in or maintain homeostasis because it's one of the central arbiters of homeostasis. It really monitors almost everything and has a hand in controlling a lot of things. So we are going to divide the nervous system up first into central versus peripheral nervous system, central nervous system being brain and spinal cord, and the peripheral nervous system is everything that's attached to it, but outside of the vertebral column. Sort of anatomically, that's how you want to think of it. Once you're outside the vertebral column, you're in the peripheral nervous system. There are cells that cross this boundary, so you can't always say that one cell is peripheral versus central, but you can say structures like nerves are peripheral. Then the peripheral nervous system we divide up into two different divisions. One is the sensory division, so this would be a sensory neuron going up into the spinal cord and eventually up into the brain. And then the motor division would be carrying signals away from the brain. So anything that's going up the spinal cord into the brain is called a sensory pathway or afferent, and any instructions coming out is carried by a motor neuron or along a motor pathway, and those are referred to as efferent pathways or efferent. I say efferent just so that you can't ever confuse it with afferent, and you do want to know those two words. Then the motor division gets further subdivided into the things that you have voluntary control over, which is the somatic nervous system, major control of the bodies. Anytime you see soma, S-O-M-A, it refers to the body in general. And then the autonomic nervous system is everything that's involuntary, as that says, like smooth muscle, cardiac muscle, glands. And then the autonomic nervous system gets further subdivided into the sympathetic and parasympathetic divisions. We are not going to go into those in this chapter. The autonomic nervous system will get its own chapter. So we're going to spend most of the chapter talking about what neurons do, but before we get into neurons, we have to mention these 6 cell types right here. So for these, you want to know which ones are in the central nervous system, which ones are in the peripheral nervous system, and what each cell does. First up is the astrocytes. These are probably the most complicated and powerful, meaning that they have like the most different functions of all of your glial cells or neuroglia. These are sometimes called astroglia because they look like asterisks or little stars. This first bullet point here where it says help determine capillary permeability, what this means is that there are blood vessels that run through your nervous system. We will talk later about a blood-brain barrier, but you don't want to think of it as this place where blood flow stops and nervous tissue begins. Nervous tissue is vascular, but there are structures in place to separate the blood from the nervous tissue around it. One of those structures is the astrocytes, and they don't really show it here, but the astrocytes wrap themselves around capillaries or small blood vessels in the brain and help to regulate what leaks out of the bloodstream into the brain. As this says here, they regulate glucose levels. This is very important in the brain because it relies on glucose. It cannot go anaerobic and it cannot go keto. It can't burn fats and proteins. It has to have glucose. And your brain controls sort of minute by minute glucose levels in different parts of the brain, both through blood flow and through the activity of astrocytes. Some astrocytes are located near synapses, places where 2 neurons communicate and they dump neurotransmitter on each other. So one of the things that astrocytes do is vacuum neurotransmitter out of the synapse, like we had acetylcholine esterase digesting acetylcholine. Astrocytes just pump the neurotransmitter out of the synapse so that signaling is only happening momentarily. They also participate in information processing, but we don't really understand that very well, so I just leave the bullet point in there. Next up are the microglia. These are a form of phagocyte, so they phagocytize whatever is not supposed to be there. Mostly debris, because it's relatively rare for microorganisms to make it into your central nervous system, but it does happen. That is meningitis. Then you have the ependymal cells. These are a little tricky to understand anatomically. You may not know it, but you have little hollow spaces in your brain. So this little thing here labeled 16 and that black spot next to it, those are your lateral ventricles. Number 9 right there is your third ventricle. So it's a little hollow part of the brain that's filled with cerebrospinal fluid, and it is lined by ependymal cells that have a vascular supply to them. This doesn't really show that, but they are incorporated into a larger structure that contains blood vessels, and they pump fluid, well not fluid, they pump solutes from the bloodstream into the vesicles and then fluid follows. So just like the cells that line your sweat glands are going to pump salt and urea into the sweat gland, and then the water's going to follow because you've generated an osmotic gradient, the same thing happens with your ependymal cells. So they pick and choose what from the bloodstream is going to get pumped into the cerebrospinal fluid, and then as they pump solutes, water is going to follow. Then you have oligodendrocytes. That is this cell right here. What you want to notice about the oligodendrocyte is this is the soma right here. This is the control center. That's where the neuron is located, and it sends out processes that come into contact with these yellow processes called axons. Those are what carry the action potentials or nerve impulses along from one part of the brain to the other. For right now, we're just going to say that the oligodendrocytes produce the myelin sheath in the central nervous system. After we talk about a different cell type, we'll mention what the myelin sheath does. But I did want to mention that these cells send out these processes because later when we talk about nerves getting severed, when you sever an axon in the central nervous system, it loses its connection to the oligodendrocyte, and then the myelin sheath can't reform, or you lose the myelin sheath. Let's see. Now we're into the two cells in the peripheral nervous system. First up are the Schwann cells. These are found surrounding the axons in peripheral neurons or peripheral nerve fibers, where they also form the myelin sheath. So here you'll notice that each Schwann cell completely surrounds just one axon. So if you were to say sever this axon right here, these Schwann cells here would still remain, even as the rest of the cell, the rest of that neuron would deteriorate. So when this peripheral neuron wants to regrow, it's got a path that it can follow to regrow. And so this is why damage to peripheral nerves is easier to fix than central nervous system damage. So spinal cord injuries are harder to fix, or really at this point still impossible to fix, because we can't get the neurons to regrow through a myelin sheath that's not there anymore. And they use that sheath as a pathfinding process or to aid them in their growth. Moving right along, then you have satellite cells. As this says right here, this surrounds neuron cell bodies in peripheral nerve system ganglia. So now we need to know what a ganglion is. A ganglion is a collection of neuron cell bodies outside of the central nervous system or in the peripheral nervous system. So if we go back to this slide right here, this is what a sensory neuron looks like. It has its dendrites maybe out in your fingertips, and then this axon right here, if this is in your fingertip, this axon is going to run, well, technically it's a dendrite, but it looks like an axon, sorry, is going to run all the way up your arm into your spinal cord. This end is going to be in your spinal cord. And the soma right there that has the nucleus is going to be just outside the spinal cord right there, in what is called the dorsal root ganglia. Not that you need to know that. But so you just want to imagine this bulge here and that bulge there is a couple hundred or more of the cell bodies of sensory neurons as they're making their way into the central nervous system. So this is one of those cells that crosses peripheral and central nervous system, because one end here is in the central nervous system and then the rest of it is in the peripheral nervous system. Okay, so now we can actually talk about neurons. What do neurons do? Really what they do is relay information or signals from one part of the brain to another part of the brain, or from the brain to a target organ, however you want to think about it. When I talk about how they work, I like to use old-fashioned telephones that run through a landline. I know nobody has them anymore. I don't even have a landline, and I'm almost 50. But so you want to imagine the way an old telephone with a cord works. The dendrites of that phone are like the speaker that you talk into. You speak words into the phone. The phone itself, in this case, in this analogy, is the soma, right? The phone takes your words and turns it into some sort of electrical signal that's then going to run down the phone lines. So the axon is the phone line, and the electrical signal in this case is action potentials. When that electricity gets to the phone on the other end of the conversation, there's a speaker, and words come out of that speaker, and that's what the axon terminal does. So you speak words into one end of a phone, Electricity happens, travels from one place to another, words come out the other end. What neurons do is dump neurotransmitter on one end of a neuron. Electricity is going to happen in the cell body and in the axon. That electrical signal, or action potential, travels down the axon, and then words or neurotransmitter come out the other end. So the same thing is kind of happening on both ends, this neurotransmitter thing. So then to briefly go over with a little bit more detail what each part of the neuron does, the dendrites are the sensory regions because they contain neurotransmitter receptors. You can also find them on the cell body, but we're going to concentrate on the dendrites. These are ligand-gated ion channels. So they are exactly the same as that acetylcholine receptor, which is a neurotransmitter, that we saw when we did the muscles. So neurotransmitters, which in this case are the ligand, are going to bind to these receptors, and then they're going to pop open, and ions are going to flow back and forth across the membrane, and there's going to be changes in the resting membrane potential. The cell body is where the nucleus is, and it's also where the overwhelming bulk of all of the organelles are. So the Golgi, the rough endoplasmic reticulum, and whatever else might be there, mitochondria. There are some mitochondria and some machinery down in the axon terminals, but not as much as in the cell body. Some books or resources you may see might have this term called Nissl bodies. I want to make you aware of it, but I don't ask that you know it because it's kind of a leftover term. As I understand it, Dr. Nissl, who developed a stain that neurons and other cells absorb, was looking at nervous tissue, which is part neurons and part glia. And because the neurons have to have enough rough endoplasmic reticulum to manufacture proteins that support the entire axon, they have much more rough endoplasmic reticulum in their cytoplasm. Their cytoplasm is just denser. It's packed with more stuff. So when you stain nervous tissue with this Nissl stain, the neurons looked darker and had chunks in their cytoplasm that didn't appear to be in the astrocytes and other glial cells. That led Nissl to believe that there was a substance in the neurons that weren't in the astrocytes or glial cells, but it was just a big difference in the density of the same substance, making the neurons appear darker and different than the glial cells around them. So now you all know more than somebody that was smart enough to get something named after himself. This is called standing on the shoulders of giants. It is one of the benefits of living today, because we have the benefit of lots of people who thought they were pretty smart in the past moving right along, and we think we're pretty smart, and we are. Sorry. But it is amazing to think that you all know more than the smartest people on the planet did a couple hundred years ago. It's just kind of neat. Sorry, next up, the axon. So that is the long process that comes off of the neuron. Technically speaking, each neuron has only one axon. Some neurons look like they might have two, but we call one a dendrite and the other the axon. It arises from this part right here is called the axon hillock, and it carries, as we said, action potentials, which are always going to travel from the soma to the axon terminals, which is the end of the axon, and that is where neurotransmitter is going to be released. They are sometimes called synaptic knobs, synaptic end bulbs, or boutons. That's more of a French term. Currently, the most unifying language for science, common language for science across the globe, is English, but for a while it was French. Everybody communicated, or at least people in Europe and in contact with Europe, like probably people in the Arabic-speaking world, would also communicate in French. Let's see, moving right along. Oh, the myelin sheath. We said we would get to that. So remember, this is both the oligodendrocytes in your central nervous system and the Schwann cells in your peripheral nervous system produce this substance called myelin. It is part protein, part fat. It's a mixture. But given that it has a fair amount of lipids in it, it doesn't conduct charge. It is an electrical insulator. And its primary function is to make action potentials travel faster. We don't really have time to talk about why they travel faster with myelin. You just want to know that they do travel faster with myelin. Without the myelin sheath, your nervous system wouldn't work fast enough for you to keep up with reality, and consciousness as we know it would definitely not be possible, because that requires a whole lot of crosstalk between different regions of the brain. Oh, that's right. I just wanted to point, I forgot why this slide was here. I just wanted to point out this one term that actually, both in the central and peripheral nervous system, you have nodes of Ranvier, but peripheral nervous system, it's Schwann cells, central nervous system, it's oligodendrocytes, but there's still nodes of Ranvier. All right, then, let's see, we're 21 minutes in. We'll keep going for a few more minutes. This being A and P, we are going to classify things because it's what we like to do. So there are 3 different structural classifications of neuron based on how many things branch off of the soma. So if you look at this neuron right here, it's got an axon and several dendrites branching off of the soma. That is a multipolar neuron. If you look at this neuron right here, it's got 2, whatever you would call them, processes. So they both look like axons. I can't speak, but one of them is going to have neurotransmitter receptors and the other end is going to have synaptic vesicles. So the end with the receptors is going to be called a very long dendrite, and then the other one, if that's where the neurotransmitter comes out, would be the axon. So this one would be the axon. And then this is a unipolar neuron. So there's just one process coming off of the cell body, and then this peripheral process, which is kind of part dendrite, part axon. I've never really thought about how to label these before, but this is what all of your sensory nerve cells look like. So this part right here, or that thing right there, would be in your dorsal root ganglion, and then one end is in the periphery and one end ends in your spinal cord or brainstem somewhere. This is what all your motor neurons look like, or most of your motor neurons, where this part would be in the brainstem or the spinal cord, and then this would go out to the target organ, muscle, gland, whatever. Then, functionally, there are 3 different classifications of neurons. One is sensory, so if information is going up and in, those are all sensory neurons. And then if information is going from the brain out, those are all motor neurons. So everything here blue is a sensory neuron, everything here red is a motor neuron. And then within these 2 pathways, there are what are called interneurons. So right here, this is labeled interneuron. I don't know if you just heard that, somebody honked their horn. So right here's our friend, the sensory neuron, is going to run into the spinal cord, actually goes up the brainstem a little bit, and is dumping neurotransmitter on this cell, which is going to relay it to that cell. So this neuron is in between 2 other neurons, so we call it an interneuron. In this motor pathway, there's just 2 neurons. This primary motor neuron and spinal cord motor neuron. I don't know if that gets a name. Lower motor neuron. So I'm going to stop this screencast here, but I want to make you aware of this. So if you start reading past this, this is how my course slides are outlined, because I think my way of covering the material makes more sense. But in the OpenStax book, and if anybody is using the Mariab book, which is really popular from Pearson, they use the same information flow, which I really don't like. So they start here with the generation of an action potential, talk about how that runs down the axon, and then releases neurotransmitter, and then they talk about synapses. I start here at the beginning for this neuron and talk about how this neuron is going to go about generating the action potential, where that comes from. Then we cover the action potential and neurotransmitter release. So I just do it a little differently. To me, this just makes more sense. I took a neurobiology course. Actually, I didn't learn about neurons till graduate school, really. And my graduate school text covered it the same way. This was first and then that, and I was really confused until I went back and read the book again. I was like, oh, the book is weird. So at any rate, that's it. I'll see you next time.